the challenge for influenza virus is that the virus change very quick. Because it changes so quick, then if you make the vaccine from one strain this year, it may work well, but then next year it may not work very well because the new strain emerge and circulating in the population. And so one way to address that is that like you have to change the vaccine or to match with the newly emerging. And like I explained a little bit before, in human, we had a committee who reviewed the data because they gather data from different parts of the world, and they make a prediction of which strain going to circulate next year.
Mm-hmm.
In pigs, it's not as organized like as in human medicine. Like we don't have the way to update the vaccine that quick. Like the vaccine in swine against the swine influenza is not updated like as frequent as the vaccine in human.
Welcome to Swine Podcast Canada, and brought to you by Wazenetics. I'm Aline Remus, dedicated to bring you the insights and latest discussions in the swine industry. Today we'll explore a topic that is crucial for swine health and production efficiency: vaccines. And we'll be talking specifically against swine influenza virus. Our guest is Dr. Hiep Vu, Associate Professor, Department of Animal Science, University of Nebraska-Lincoln, whose expertise spans from vaccine development, viral pathogenesis, and strategies to control one of the most dynamic and challenging diseases in pigs. Welcome to our podcast, Dr. Vu.
It's my pleasure.
So, Dr. Vu, could you tell us a little bit how you got to this field? What passionates you in working such a challenging but important field?
I grew up in a swine farm, so I helped my parents managing a small farm, and we face infectious disease very frequently, and whenever the disease emerge or appear in the farm, like it causing devastating effect. And since I was very young, I was exposed to the concept of vaccination is the tool to control infectious disease, and I've been interested in then developing vaccine to control infectious disease in animal, particularly pigs, because I grew up with raising pigs.
And you did your studies in veterinary sciences or biological science?
Oh, I did my study in veterinary medicine. So I received a degree of Doctor of Veterinary Medicine in Vietnam before I moved to Nebraska, where I did my master and Ph.D.
Oh, that's fantastic. So that we are all in the same page, could you tell us what is a vaccine and what make one vaccine different to another?
A vaccine, so different way to define it, but it is a substance where we use to inoculate to animal, obviously, and human to induce immunity against a specific disease that we want to prevent. For example, we develop vaccine against COVID, right? Because we want to prevent COVID in human. So vaccine used for animal as well as human, and there are different kind of vaccine. One of which is that we just simply grow the pathogen and then inactivate it and use it as an inactivated vaccine. So we grow the pathogen up, inactivate it, and inoculate to the recipient. The idea is that the immune system will recognize the killed agent, and we train the immune system so that when in the future, when it encounter the infectious agent, then the immune system is ready to fight against it. So that is the very classical way to make the vaccine inactivated. The other way commonly used is we call live attenuated vaccine. So instead of using inactivated agent or pathogen, we use a live, but we weaken it. We make it weak so that when we inoculate to the animal, the pathogen do not cause disease to the recipient, but it will replicate and it will induce immunity. So that is the second kind of vaccine, live attenuated vaccine. And we use that, there are quite a few live attenuated vaccine for use in human as well as animal. And then the next group of vaccine, I call them in general, I call them subunit vaccine. So instead of using the whole pathogen, either live or inactivated, we use only a portion of the pathogen to induce immunity. And because we use only a small portion of the virus, and therefore we call it the subunit vaccine. So an example I've been using to explain the different to my student, different kind of vaccine to my student is that, like I said, that if I want to introduce me to my student, right, I can give the photo shot of me from head to toe, and the student, when they see my photo, they know, okay, this is Dr. Vu. But the other way is that I don't give them the photo of me from head to toe, but instead just the head shot. And my student, when they see me, they will recognize this me. They don't need to see the whole body. They don't need to see the image of the whole body to recognize this is Dr. Vu. They just need to see the critical part of my body, which is the face, and they recognize it's me. So subunit vaccine is similar, like you give the animal body the headshot. That is just like the critical component of the viral pathogen that help the immune system recognize the pathogen.
So I can understand, what is the vaccine that is currently used in swine, and which type of vaccine you work with?
For swine influenza virus, we currently use the inactivated vaccine. But like in swine, we may use multiple different vaccines depending on what disease we want to protect against. For example, like influenza, mainly we use inactivated vaccine. But if you talk about another disease of swine, for example, toxin reproductive and respiratory syndrome virus or PRRS, people may use live attenuated vaccine. So different vaccine for different disease.
And how are they performing in the field? What are the factors that will influence the efficacy in protecting pigs from the against the circulating strains of
swine
influenza? That we have.
So for swine influenza, like the critical factor that affecting the efficacy is the matching between the virus that you use to make the vaccine and the virus that the pig will encounter in the field. So the closer between them, the better the efficacy. And swine influenza is quite similar to seasonal human influenza. Like in human, we had a committee who would do the surveillance of the virus circulating, and they make a prediction of which strain of influenza virus will circulate the following season, and then they prepare the vaccine. And from time to time, like the prediction is not like as accurate as we wish, and then accordingly the vaccine efficacy is not as good as we expect. And so the same thing happened with the swine influenza. So the closer between the virus that we use to produce the vaccine and the strain that's circulating in the field, then the better the efficacy.
So do I get right that often the strain changes even from one season to the other, to 1 year to the other, to better match what we are seeing in the field?
Yep. So the challenge for influenza virus is that the virus change very quick. Because it changes so quick, then if you make the vaccine from one strain this year, it may work well, but then next year it may not work very well because the new strain emerge and circulating in the population. And so one way to address that is that like you have to change the vaccine or to match with the newly emerging. And like I explained a little bit before, in human, we had a committee who reviewed the data because they gather data from different parts of the world, and they make a prediction of which strain going to circulate next year.
Mm-hmm.
In pigs, it's not as organized like as in human medicine. Like we don't have the way to update the vaccine that quick. Like the vaccine in swine against the swine influenza is not updated like as frequent as the vaccine in human.
What are the tools that we actually have to try to match the vaccine strains with the regional or herd levels virus strains that we see? And what are the strategies that are available to us?
Nowadays, sequencing technology is very, very popular and affordable. So the sequencing become more and more cheaper than the past. The swine farmer operation, they whenever they experience respiratory problem, and they may collecting the sample and they do the diagnose and they can sequence to see what is the sequence of the strain that's circulating in their production. And we had a very good database so that we can tell how close and how far apart the virus that affecting our farm compared to the one that is circulating right now.
What your research is telling us about that?
I did not. My research is not directly related to surveillance. I do not do the surveillance, but instead, I try to develop the vaccine platform that allow us to update quicker. Remember, we said that because the virus mutate, the virus change so quick, and we need to have the way to produce the vaccine more effectively in the sense that we can update it faster. Because nowadays we have the sequencing technology, we can sequence and we can identify new virus very efficiently. So then we need a platform that allow us, once we know the sequence of the new virus, we can use that to formulate the vaccine faster and safer compared to the way that we do in the past.
Can you tell us a little bit more about this platform?
So here's how it's different. In the past, if I want to produce the influenza vaccine, then I may go to the farm that broke with flu, right? I collecting the sample, bring it back to the lab, and then try to grow the virus or isolate the virus and amplify it. And once I amplify it, and I have to inactivate it. Remember, I start with the infectious virus because I culture the virus up, and then I perform the inactivation. And I have to be very sure that I inactivate it completely. If I did not inactivate completely, and then I use that one to inject to the pigs, then potentially I may induce disease in the recipient.
Right.
But with the platform that I'm working right now, I do not use the whole virus to immunize the animal. We know that the virus had the surface protein, just like me, with the headshot, right? I use the surface protein of the flu. Just one piece of the protein. We don't use the whole virus particle. And so then I take that gene, I clone it into the plasmid, and I call it the DNA vaccine. So it's somewhat similar to mRNA-based vaccine. So we inoculate the animal only with the DNA encoding the surface protein of the virus. And so we do not inoculate the animal with the infectious agent, just a piece of DNA encoding the surface protein. So when the DNA go to the pigs, it will be transcribed to produce messenger RNA. And then the messenger RNA will become the protein. And the protein is the one that stimulates the immune response. So it's non-infectious and it's safer. And also it's faster to produce.
So can we also say that is you said it's a safer vaccine. We can trust this type of vaccine because we're almost sure that we will not induce a disease, exactly, because it is not an inoculate virus. So you don't have that problem of thinking, well, data is really inactivated or not. You're just giving a snapshot of let's say the as you said, a great example, the picture, that's just the face of this virus, and then the immune system will respond. So there is anything that you can do to boost the effect of this vaccine?
So the approach that I mentioned to you, the scientists have been tried, I would say 30 years already. But there is limitation of that. The key is that the DNA piece that we inject to the animal, it have to enter the animal cell, and then it could be converted into protein. So if the DNA cannot enter the animal cell and be converted into protein, then it won't stimulate the antibody response. So the limiting factor is that how we introduce the DNA molecule into the animal cell. And so what I do in my lab is that we use lipid nanoparticle. So we wrap around the DNA plasmid with the lipid. And the purpose of that is the lipid nanoparticle will enter when we inject the animal with the vaccine, then the lipid nanoparticle will help mediate the entry of the DNA into the cell. And once the DNA enter the cell, then it will be converted into the messenger RNA, and then it will be converted to protein, and the protein will stimulate the immune response. So different people have tried different approach how to enhance the entry of the DNA into the cell. And in our lab, like the key is that we found a lipid nanoparticle formulation that allow it entry effectively, and consequently we get the very robust immune response. I like to explain a little bit different why it is critical. Imagine right now, like you may hear about the highly pathogenic avian influenza, right? Highly pathogenic avian influenza, like in the past, like it infect avian. That's why the name come from, highly pathogenic avian influenza. But then, like last year, we found that it infect dairy cattle. The virus have the ability to change the host range. Instead of infecting only the poultry, now it infecting dairy cattle. And we worry that it may infect pigs, for example, because pigs can be infected with this highly pathogenic avian influenza virus. Because it is highly pathogenic, then by regulation, like you cannot culture the virus under the regular biosafety level 2 laboratory. You have to have the right biocontainment, which is the biosafety level 3, if you want to culture that virus. And therefore, like because it highly pathogenic, it is risky when you handle the whole virus. But in my case, like I just handle only the DNA, the piece of DNA molecule. Like I don't even handle the virus, so I can start formulate the vaccine under the standard biosafety level 2 environment, and so it's safer.
Yeah, you touched in the subject, but what are the advancements or innovations in swine influenza vaccination that are on the horizon and can come to also benefit the Canadian swine industry?
So like I said, like flu, it's changing quick, and in order to control that, is that we just have to find a way to survey the virus out there. Like what are the virus currently circulating in the field, and surveillance is a key part of that. And like I said before, like sequencing become more affordable. So the more you sequence and the more surveillance you do, then the better knowledge you get about the landscape of the virus that circulating in the Canadian swine population. And then with the technology nowadays, like allow, for example, the DNA vaccine that I'm mentioned here, allow you to custom made the vaccine for the operation. Because I believe Canadian is similar to the US, where you have several large operation, right? And then you can custom made the vaccine fit specific for this operation, because we had the sequencing data knowing what strain of the virus is circulating in this operation, and we can make the vaccine match to the strain, and so it enhance the efficacy of the vaccine. And because the vaccine is safe, because the vaccine is non-infectious, and we can update it quicker, because obviously the virus changed. So maybe this year the sequence is like this, the strain is like this, but next year you may have a new sequence, and then you can update the vaccine to match that new virus.
So can we say that the key for the success in using this type of vaccine is really that our population vets keep an eye on, keep monitoring, they sample, they really do this continuous monitoring of the disease and sampling. I get what I'm getting, like a take home from you is really that is really important to always sample to see what we have.
Yep, the best the first step to control anything is that you have to know where they are, and what kind of pathogen affecting the farm, right? Because if we don't have that piece of information, it's going to be difficult to design a protocol to control it. So active surveillance is the good way to manage the farm.
We always say in modeling that you cannot model what you don't know, right? And I think that really applies here. You cannot produce vaccines to things that you're not monitoring, that you're not sampling. So it's very important to keep an eye out there.
Yes, especially this virus because it changed very quick.
It's time for our famous three.
Why Genetics turns podcast airtime into brand authority. We don't sell ads, we elevate voices. Curious how far your voice can go to become a reference in the industry and attract more leads? Scan the QR code and discover how we can turn your expertise into unmatched brand authority. Let's transform expertise into influence, starting now.
So we're going towards the end of our episode. So if you at home are liking this episode, please hit subscribe, give us a like, and write comments below. You can ask questions to Dr. Vu. And if you have questions that were not addressed here, maybe we can invite him to come back and answer us some more questions and discuss, because this is a very interesting topic. Dr. Vu, I will go for our three final questions, if you don't mind. So could you tell us what is your favorite swine-related book or resource?
Book swine specific or related, I don't have a book about that, but like in terms of research, like I do like the surveillance, like I described before, especially with the sequencing technology, so that we know what are the pathogens circulating in the operation or in the country or in the region. I thought that is an important part of the research. It must be done continuously. And then I like vaccine development. That's what I'm doing. So we try, as a scientist, like my goal always like to research and develop the new kind of vaccine that is safer and more efficacious than the previous generation of vaccine. Safer, more effective. But in terms of book, if you want to read like non-swine related for general understanding of vaccine and how like important vaccine is in terms of control disease, like I like to introduce the book about polio. By the way, Eileen, like do you know what is polio? Now I'm interviewing you now.
I'm all for it. I know that I was vaccinated for it, but I don't know much about it. No.
Yeah. So the reason why like the generation like us, like you and me, we don't know much about polio, or we do not see people with polio, is because of the vaccine. Like
Right.
when I'm asking my student, and some of them like have no clue what is polio because they do not see it. They haven't seen the case of that. But if it was like about 100 years ago, you may see more. Since the time we had the vaccine, we used the vaccine, and really like we reduced the incidence of polio significantly, to the point that like we rarely see a case of polio. And that's why like people don't even, some of the younger generation don't even know what it is. But in order to go from like a lot of cases to like almost like no cases nowadays, that is a process. And vaccination play a significant role in that. If you like to read about some topic, because the pathogen do not care whether it's human or a pig, right? It in fact,
Right.
and so it kind of applicable. So I like to introduce the book called Polio, an American Story.
Awesome.
It's non-scientific. It's very layman language. It's very nice reading. I really love it. The reason why I love it because like the entire like country campaign in terms of money put into their research and how they work to develop the vaccine, and after the vaccine is developed, how they use, because you may develop a very good vaccine, but if the public does not accept that vaccine, they do not like to use it, that vaccine won't be like any good.
Could you repeat the name of that book for us?
It's called Polio, an American Story.
Fantastic. I'm really looking forward to read it. It's like I always want to understand more about the topic and how we get to the point we got, because we take everything for granted. We take, I think polio is a great example. Like, yeah, I know I got a vaccine, but I have no idea what is it, right?
Well, back to the about 1920 up to that point, like there are many healthy, like wealthy, like family. They try all means to prevent the kid from being affected by the disease, but it's just helpless because they don't know how to prevent the the kid from getting the virus. And then thanks to the vaccine, then like nowadays, like we don't even really like know it exists, because like, a lot of young kids, they don't even know about polio. And the vaccine, like I said, vaccination is the big contributor to the success. And in the book, they would discuss how they campaign to raise fundraising and to put into research to develop the vaccine. And then after the vaccine is made, like how it's used. Because back to the day, I believe like they go to the school and they give the vaccine to the kid at school. But it's a success story.
Just to finish our last question, I could stay longer here just talking, ask you a question about vaccines, but unfortunately our time is limited. So could you just give me, in your opinion, what sets a successful swine professional apart from those that are not?
Well, I may say that more in terms of research, because that's in my field. Because in terms of production or practice, that may be different. But in terms of research, and I think like staying focused is the key. Keep like doing what we believe is important, and you may face some challenge, but like stay focused, stay like commitment to that, and try to solve the problem.
Well, Dr. Vu, thank you so much for joining us. That concludes another episode of Swine Podcast Canada, presented by Wazenetics. Thank you so much for joining us today. Do not forget to subscribe to our podcast to stay updated to the future episodes. And if you enjoyed today's discussion, please leave us a review. Your feedback is very important to us. Until next time, this is Aline Remus signing off.
Thank you so much for having me.